Hello, today we're going to have talk number 11 in our Principles of Biochemistry series, 7th edition, Albert Lehninger author. Today we're going to look at cooperativity and continuation of our discussion of ligands, which again are moieties which react with proteins in a sequential manner, and the equations and mathematics that we use to help describe this process, which is in the form of hemoglobin, the addition of one followed up to 4 oxygen atoms, each of which is bound to one of the subunits, whether alpha or beta. So to help quantify this process, we look at the Hill equation. The Hill equation, mathematically speaking, is defined as the log of the ratio of y divided by 1 minus y, where y is the compound that is being formed. And this is equivalent to the number n, which is either 1 or greater than 1, multiplied by the log of the ligand's concentration minus the log of the dissociation constant. So under conditions of N1, you have no cooperativity. There's no positivity nor negative tendency of a ligand to bind. And then positive cooperativity refers to a molecule such as hemoglobin, where the initial oxygen is most difficult to bind, and the following oxygens bind more readily as the hemoglobin changes its conformation to allow further and further binding of oxygen to occur. The remaining protons and carbon dioxide are dissolved as bicarbonate and dissolved carbon dioxide, and these have an impact on the conformation. Hemoglobin binds a proton to amino acids, and the carbon dioxide binds to amino terminus of subunits. And these are elements which help configure the hemoglobin molecule as the oxygen binds. Hemoglobin modulators, one of these is 2,3 bis phosphoglycate, and this reduces hemoglobin affinity for oxygen. This is reduced at high altitudes, so you get more affinity for oxygen at higher altitudes. This compound actually decreases. In sickle cell anemia, valine replaces glutamate at the beta subunit position 6. So when you have the valine replace glutamate, you have a loss of the charge of glutamate, and you have an aliphatic nonpolar valine present. So you get a hydrophobic area occurring, a conformational change occurs, and as a consequence, a person can get with sickle cell anemia, a disease with sickle cell trait. Only one of the 2 genes is affected, and you only get clinical symptoms occurring if you've got a strenuous situation, hypoxia, or rigorous exercise. But if you have sickle cell anemia, you're under that particular stressor at all times. The immune response is an example of a ligand interaction where you've got the ligand as a separate protein that occurs, and in a humoral system, this is geared towards bacteria and extracellular viruses modulated by B cells, and cellular immunity is geared towards parasites, foreign tissues, and intracellular viruses modulated by T cells as broad categories of response. And the ligand is referenced in the cases that I've enumerated. So when you look at the immune response, and this is kind of a shorthand, you have macrophages, which ingest large particles in cells by phagocytosis. The B lymphocytes, the B cells, produce and secrete antibodies. T cells, in broad categories, you have cytotoxic killer T cells, which interact with the infected host through receptors on the T cell surface, and helper T cells, which interact with macrophages and secrete cytokines, which are interleukins, and they stimulate the T cells, the helper cells, the cytotoxic cells, and the B cells to proliferate. The immunoglobulins have variable and constant domains. The variable domains have recombination capacity. and they comprise a part of the immunoglobulin. So by and large, IgG is the recovery phase, is the most numerous of the, in terms of vast concentration, is the most numerous of the various classes of immunoglobulins. You have IgM, which is initial antibody response. So the antibody response in the immune case is IgM followed by IgG. IgA is found in secretory situations. It's a monomer, dimer, or trimer. IgE is found in allergic responses, and IgD is not really something we need to discuss at this point. It's also a dimer. So small molecules are generally not anergetic, but a hapten can be a small molecule combines with a larger molecule and become anergenic. So if you look, for example, at IgG, you've got the variable parts at the end and the constant parts at the proximal end of the Y arm, and then the base is there. There's a papain cleavage site in the center here, and this is the structure of it. You have the constant, the Fc and the FAB parts, which are domains on the immunoglobulin molecule. Amino acid terminus in here, interactions at this end, antibody, the antigen binding occurs at this point in the variable area here. The dissociation constant is low for antibody, implies strong binding, and the binding strength is useful in lab techniques. So you can link antibodies, for example, to a matrix. You can incubate and bind the antigen, label the product, and do an assay. And this is used very commonly in biological systems. So, for example, you can have a case here where you can coat a surface with sample antigens, which are here. You can block unoccupied sites with nonspecific protein, which are here. You can incubate with a primary antigen against specific antigens here. And then a secondary antibody enzyme complex can bind to that antibody. You can use washing steps, so you end up getting this reaction with a marker here, and then you've got a substrate that is present, and then the substrate is activated or changed to become a detectable moiety here. So it can be a colored product, it can be a light reaction like a luminase. This is how you quantitate. These are how enzyme reactions can be utilized, or antibody binding can be utilized to detect any type of structure. So if you quantitate antibodies, you can characterize them this way and use this to get a purification of results. And we call this ELISA, enzyme-linked immunosorbent assay. This is what it is, and this is used a lot of times in biological systems to help quantify an immune response. Western blot is an example where the substance itself is subjected to electrophoresis separation, labeled specific antibody, and the antibody is linked to a substrate or enzyme. So the substance in the previous slide would be at the base level, and that's when you subject to electrophoresis. You can work within the matrix to get this result. For chemical engineering and protein interactions, we look at larger molecules. So these proteins are involved intimately with movement of macromolecules, muscle, flagella, microtubules, and membranes. These all have movement associated with them. Myosin is a part of muscle with thick muscle filaments. Actin is thin muscle filaments. And there's a myofibril matrix that helps encompass both of these and allows these reactions. It's more of a sliding method which occurs within the filament, where the filament thickens or thins out depending on the presence of myosin actin interacting with each other or not interacting with each other. Myofibrils are surrounded by sarcoplasmic reticulum, and the entire unit is a sarcomere. We'll be discussing muscles at a later point. Their regulatory proteins are involved in this process. Tropomyosin blocks myosin binding to actin, and troponin, which is a marker in myocardial disease, myocardial injury, a very specific protein involved with that organ, binds calcium, which is released from neural impulses. It allows for active binding to myosin. So the presence of troponin and various forms in circulation is indicative of myocardial disease. And that's it for this talk. Thank you so much.